The overall goal of this procedure is to induce compulsive-like behavior in rats for the study of obsessive-compulsive disorder. This is accomplished by first training rats to collect a food reward preceded by a compound stimulus consisting of a light and tone. The second step is to train the rats to lever press for the food reward, which is accompanied by the compound stimulus. Next, the signaling properties of the compound stimulus are attenuated by repeated exposure without the food reward and lever presentation. The final step is to measure lever press responses following signal attenuation. Ultimately, the signal attenuation procedure leads to an increased number of lever presses not followed by an attempt to collect the food reward.
The main advantage of this technique over existing methods like marble bearing, T-maze alternation, etc. is that it is highly validated, highly selective for anti-compulsive drugs, differentiates between compulsive-like behaviors and behaviors which are repetitive but not compulsive, and uses measures which are quantitative, unbiased, and unaffected by inter-experimenter variability.
The implications of this technique extend towards therapy of OCD because it can be used for detecting anti- and pro-compulsive effects of novel pharmacological and non-pharmacological manipulations.
The first step is to handle the rats for about 2 minutes a day for 5 days prior to the beginning of the study.